Our next session is going to be another In the Pipeline series where we look at some exon skipping therapies that are using some conjugates to help aid in delivery. So first we're going to hear from Dr. Scavina, and then we'll introduce the panel.
Dystrophinopathies, which include Duchenne and Becker muscular dystrophy, are caused by a variant or change in the DMD gene, resulting in little or no production of the dystrophin protein. These changes can include deletions, duplications, or nonsense mutations that disrupt the reading frame, which is the instruction the body follows to make a protein. When the reading frame is disrupted, it prevents the production of that protein, in this case, dystrophin. Let's look at the reading frame as a sentence. DNA provides the instructions for the protein, essentially like the letters in a sentence. Depending on which letters are missing or duplicated, the sentence may be completely changed and not make any sense. Or it may make sense, but not exactly like the original sentence. Therefore, the instruction to make the protein is altered, and either no dystrophin or reduced dystrophin is produced. This is seen with deletions, duplications, or smaller point mutations. If letters are missing, called deletions, exon skipping allows some letters that border the original deletion to be skipped over to correct the sentence and allow some protein to be produced. Antisense oligonucleotide, or ASOs, are single-stranded pieces of DNA that are used to restore the reading frame through exon skipping. If we can restore the reading frame or correct the sentence, we can produce dystrophin again, although it may be modified. The protein may be smaller and the amount may be less than typical. New technologies are being developed to aid in the delivery of exon skipping therapies such as ASOs. These newer technologies act as helpers to the ASO to increase the targeting and delivery of the ASO to muscle itself. This could potentially increase the amount of drug delivered and increase the amount of protein produced.
All right, we're now going to be joined by our panelists for this session. So joining us today, we have Elizabeth Ackerman from Avidity Biosciences, Ash Dugar from Dyne Therapeutics, James MacArthur from PepGen, and Elizabeth Smith from Sarepta. So if you have questions, please feel free to submit them to the comment box below. We're going to have a Q&A after we get through all the presentations. And with that, we're going to hear first from Avidity Biosciences. So Elizabeth, you can take it away.
Great. Thank you so much, Eric and Ryan and PPMD, and for the opportunity to be part of this great panel. And I've been really enjoying the conference. My name is Lisa Ackerman, and I'm the therapeutic area head at Avidity. Next slide, please. This is our forward-looking statements. Next slide. So our mission at Avidity is to improve the lives of people affected by serious diseases by advancing a new class of targeted therapeutics. And I just want to take this moment to say that we are truly honored to be able to serve this community. And we know how important it is that we understand each of your personal perspective and your personal journey with DMD for us to better help serve you. And we are also incredibly grateful to PPMD and other patient advocacy groups that enable companies like us to make these deeper connections with your community. Can I next slide, please? So you just heard from Dr. Scavina that DMD is often caused by deletion or mutation in the dystrophin mRNA that disrupts the reading frame. This does not allow the cell to make any dystrophin. Some of these mutations can be restored, have the reading frame restored by skipping a specific exon in the dystrophin RNA. When this happens, the cell makes a slightly shorter dystrophin protein, but one that is functional. So at Avidity, we are developing 3 different drugs to treat DMD, and they all work by skipping an exon in the dystrophin RNA. The first one is called AOC044, and it works by skipping exon 44. Now, we hope to take this drug into the clinic next year in 2022. The second drug skips exon 45, and the third one, exon 51. So taken together, these three drugs, we can address about 30% of the DMD patients who have mutations amenable for exon skipping. Next slide, please. So at Avidity, our drugs are made with basically two building blocks. One is an antibody, and one is an oligonucleotide, or a PMO. A PMO is a short piece of DNA, single-stranded, that can work in the cell by causing the cell to skip a specific exon in the dystrophin RNA. Now, there are 4 approved PMOs to treat DMD, approved by the FDA. And these drugs have some limitations in that they can't get into the muscle and the heart cell very well. So at Avidity, we've tried to solve this problem by taking the PMO and linking it to an antibody. Now, the job of the antibody is simply just to take that PMO and get it inside a muscle and a heart cell. Now, antibodies are naturally occurring proteins that are made by your body to fight infections, but they're also commonly used as drugs. And the reason is that they have a property to be able to specifically target a single protein. Can I have the next slide, please? So this slide shows a little bit more how our technology works. We've taken an antibody that binds to a receptor that's found on the outside of a muscle or heart cell called the transferrin receptor. When that antibody binds to the transferrin receptor, the receptor pulls the antibody and the PMO into the cell. Once inside the cell, the PMO is released and it can do its job. So its job is to find the dystrophin RNA, bind to it, cause it to skip an exon, so it restores its reading frame, and therefore more dystrophin protein can be made. Next slide, please. So I'm just going to show you one slide of preclinical data that was created by our researchers. In this experiment, the researchers took the MDM mouse model, which is a mouse that has a mutation in its dystrophin gene that can be fixed, the reading frame can be fixed by skipping exon 23. So what the researchers did is they divided these mice into 3 groups. In the first group, they gave saline or a placebo, and that's shown on the bar on the far left where you can't see it very well. The second group, they gave a PMO. That PMO was able to skip exon 23 of the dystrophin RNA, and that's shown by the blue bar in the middle. And then in the third group of animals, they gave that exact same PMO, but this time it's attached to the transferrin antibody. And that's shown in the green bar on the right. And the taller the bar, the more exon skipping was observed. So you can see that there was about a 50-fold increase in exon skipping when you attached the transferrin receptor to the PMO. So the transferrin antibody. So the antibody did its job. It delivered the PMO into the muscle cell, and the PMO did its job to make exon skipping occur. So I can have the next slide, please. So at Avidity, we're super excited about the potential to use our technology to treat DMD. And we're planning to take our first drug into the clinic, AOC 1004, that will skip exon 44 next year in 2022. Now, our drugs will be given by IV infusions. And for these programs, we think it will be about once monthly. And we will continue to partner with PPMD and the DMD community to give you information as we have it available. But we would love to hear from you, and please reach out with any questions or comments that you have for us simply by emailing patients@aviditybio.com. Thanks so much, and back to you, Ryan.
Thank you, Elizabeth. I'm going to just throw one question at you for clarification on your antibodies targeted to TFR1. You mentioned that. Can you describe what the distribution may look like? Is it just getting to skeletal muscle, or are you hopeful for other types of muscle?
Yeah, so we have shown that by using this antibody to transferrin, that we have really good delivery of the oligos that we're delivering to skeletal muscle, smooth muscle, and heart tissue.
Great. Wonderful. Thank you for that clarification. We're going to move on next to Ash Dugar from Dyne. Take it away, Ash.
Hi, thank you, Ryan. Hi, I'm Ash Dugar. I'm the head of Global Medical Affairs at Dyne Therapeutics. First, I want to thank PPMD for the opportunity to share Dyne's vision with you today. I always feel privileged to have the opportunity to speak directly with the communities and families living with the diseases for which we're developing treatments. Next slide, please. So at Dyne, our mission is to bring life-transforming therapies to patients with serious muscle diseases. And at the forefront of that is our goal to advance treatments for Duchenne muscular dystrophy, which, as we all know, is a condition that still has profound unmet needs despite the current available treatments. Our mission is truly core to who we are and what we do every day. Next slide, please. Well, you've heard a great deal around PMOs and exon skipping in Elizabeth's talk and in the talk before that. So you've heard a lot of great information already that I will try not to repeat. As you all know, patients with Duchenne don't make dystrophin because of a mutation of the DMD gene. And PMOs use a mechanism called exon skipping that you've heard about to skip over the sections of genes with the mutation that cause Duchenne. One of the biggest challenges to having effective therapies is the ability to deliver enough of the PMO into muscle tissue to drive robust exon skipping and adequately increase dystrophin. And it's a challenge that Dyne is committed to overcoming. Next slide, please. So this slide provides an overview of our technology. Our FORCE platform includes a specific delivery mechanism called a Fab, or an antigen binding fragment, which is a small section of a monoclonal antibody. And we've specifically engineered to bind with a high affinity to a receptor called the transferrin receptor 1, or TFR1 receptor. And this receptor is highly expressed across the surface of muscle cells, as you just heard. So the Fab acts as a kind of key that unlocks the TFR1 receptor and allows our PMO to enter cardiac, skeletal, and respiratory muscles. We link our Fab to the PMO with a linker. Once the Fab brings the PMO to the muscle cell, enzymes inside the cell cut the linker, and the PMO is free to drive exon skipping to target the genetic basis of Duchenne. So FORCE uses the Fab to drive binding to the TFR1 receptors on the surface of muscle cells. And we selected Fabs because of advantages such as their small size, which helps with distribution to muscle tissue. And we can also redose with this technology. So this approach is intended to take advantage of specific muscle biology, allowing targeted delivery of the PMO and potentially avoiding off-target effects such as in the liver and kidney. As you've heard, currently approved exon skipping PMOs have been shown to produce very modest amounts of dystrophin. And our approach is designed to much more effectively and efficiently target PMOs to the muscle tissue to enhance therapeutic benefit. Next slide, please. So we've generated exciting preclinical data in Duchenne. This slide summarizes the power of the FORCE platform to effectively deliver the PMO to muscle, resulting in substantial exon skipping and increased dystrophin expression versus naked or non-targeted PMO in a Duchenne mouse model, the MDX model. So this mouse model has a mutation in exon 23 that interferes with dystrophin production. These mice can't run as well, have higher CK in their serum due to muscle damage, and have a shorter lifespan than a wild type or healthy mouse. This mouse model enables comparison of our FORCE platform, which you see in the blue squares, with published data from a naked PMO in the red triangle and a stereopure antisense oligonucleotide in the orange triangle. 2 weeks after a single dose of 10, 20, or 30 milligrams per kilogram with FORCE, we observed substantial exon skipping in the quadriceps, up to 66% after a single dose of FORCE. In addition to the quadriceps, we looked at respiratory and cardiac muscles. We all understand how critical treating the diaphragm and heart is for Duchenne. And as you can see for diaphragm and heart on this slide, there is very modest exon skipping using other modalities. In comparison, the FORCE platform achieves very robust and meaningful exon skipping. This increase in exon skipping resulted in the dose-dependent increase of dystrophin protein expression that you see on the right side of the slide. Our highest dose of force resulted in 8 to 10% restoration of dystrophin production with no demonstrated dose-limiting toxicities. In contrast, this naked PMO restored around 1% of dystrophin with a single dose. So this data gives us confidence that we can leverage the TFR1 mediated cell entry to drive the exon skipping PMO into muscles and generate significant levels of dystrophin protein. Next slide, please. So this functional test in MDX mice shows that the dystrophin is actually working to provide the support muscles need. This is a running wheel test which measures how active mice are throughout a given time period. The y-axis is distance traveled and the x-axis is the time of day. And between the two vertical dashed lines is the dark period when mice, which are nocturnal, run on the wheel. The gray circles are wild type mice. The black squares are MDX mice given a placebo. You can see the difference in activity. The MDX mice don't move as much. Force in the blue triangles restored function in the DMD mice compared to wild type mice. And this was not at a single time point, as these data show activity over a 12-hour period. We believe these results indicate a treatment benefit in skeletal and cardiac muscles, since MDX mice treated with force are more active over this 12-hour period compared with placebo-treated and naked PMO-treated MDX mice. So we're really excited to do additional non-clinical studies and enter into clinical trials. Our programs that we're progressing are 51 skip-amenable patients first, and then 45, 53, and 44 skip-amenable patients. Next slide, please. So at Dyne, your voices are very important to us. We have a deep commitment to understanding the community of people living with muscle diseases and Duchenne in particular. We've initiated many programs involving community webinars, conference presentations, fireside chats, patient meetings, and other outreach to understand the disease better directly from the patient community and gather input to incorporate into our drug development efforts. Next slide, please. This is my last slide. I want to thank you again for giving us your time and attention today. And I want to thank PPMD for giving us the opportunity to present our program to all of you and for this great Congress. When Ravi, Beth, and Diana, who are pictured here, and others speak to us and share their perspectives, it means the world to us. And we greatly appreciate their candor and their insights. Lastly, I want to end by saying again, it's a privilege to have the chance to speak to you. You give our work and a significant part of our lives great meaning. So thank you.
Thank you, Ash. One quick follow-up question. I don't know if you mentioned this. Would this? Are you thinking this is going to be weekly, or will it be twice monthly? What are your thoughts right now on that?
Yeah, thank you very much. So we anticipate this dosing being no more frequent than once a month. So monthly is currently what we are aiming for, and hopefully we can get to a point where it's even less frequent.
Great. If we can decrease that burden, it will be huge for the families. Absolutely. Wonderful. Okay, we're going to now transition to James from PepGen. James, take it away.
Thank you. Thank you for the invitation to speak to everyone here. We've spent a tremendous amount of time thinking about patients and how we can best treat them. And the approach I'll tell you today is, we think, a very novel approach to be able to improve the efficiency of delivery of therapeutic oligonucleotides safely to patients. If I could have the next slide, please. And next slide. So we've already had a description around the approach of exon skipping to go and essentially skip over problems associated with mutations found in the original coding sequences of the dystrophin gene. And what we're doing here, next slide. What we're doing here is taking these similar oligonucleotides, as been described already and that all of us are developing, but find a novel way to go and deliver them to cells of patients so they can do this therapeutic benefit of skipping over the deletions and mutations in the gene. Next slide. And that's sort of highlighted in the small diagram in the bottom right. That blue sequence is the peptide that PepGen has spent over a decade developing, that has an extraordinary ability to get into all types of muscles in the body and do so safely. Next slide, please. Now, we've generated a lot of mouse data associated with this approach and can go and demonstrate the skipping of exon 23 and the production of dystrophin protein in mice. Mice, unfortunately, have some limitations, however, in terms of their ability to go and predict how well it's going to perform when we ultimately move forward into patients. So we've done a tremendous amount of testing at PepGen using non-human primates as a model, which are a much better model for being able to identify whether or not you'll see efficacy and good delivery with this approach. In this particular study, we're looking at a single dose that was delivered by IV infusion into monkeys. And then we went and looked at different muscle groups: skeletal muscle, the tibialis anterior, diaphragm, and heart. In each of these tissues, we can see extraordinarily good levels of exon 51 skipping, as indicated by the higher bars, at all of these doses, 20, 40, and 60 mg per kg. We were particularly pleased to see this tremendous efficacy in both the diaphragm and in the heart. As everyone knows, the heart and diaphragm in particular are two tissues that we have to see better levels of exon skipping, which will produce more dystrophin protein to have a really dramatic impact on patients moving forward. Next slide, please. This is looking across a wide range of different muscle tissues. On the left-hand portion, different skeletal muscles, in the middle, cardiac tissue, then smooth muscle, and CNS on the right-hand side. As you can see, looking across all these different muscle groups, at all these therapeutic doses of 20, 40, and 60 mg per kg, and that's following a single dose, we see really terrific uptake of the therapeutic oligonucleotide into these muscles. And that's really important if we want to see a really dramatic increase in terms of dystrophin production. Next slide, please. So what I've shown you so far has been a single dose study, but we've also done multi-dose studies, as this will be viewed as a chronic therapy where we anticipate dosing patients once a month. In this particular study, to sort of accelerate things, we looked at once every 2-week dosing and across a wide range of doses. And as you can see here, even at doses as low as 5 mg per kg, and this is not just PMO dose, this is the dose of our drug, we can already begin to see meaningful levels of exon skipping in biceps and quadriceps and diaphragm. As we go to higher levels, we begin to start seeing it in the heart as well. And we can go and dose at these levels safely and efficiently to go and deliver the therapeutic oligonucleotide. And we think this could have a really dramatic impact. We anticipate that at therapeutic doses, we could be producing double-digit levels of dystrophin protein in patients going forward. And that would have a terrific impact in both improving ambulation and life expectancy in the patients. Next slide, please. Now, exon 51 skipping approach in DMD is our first clinical program. It's the focus of the company. And we're currently in preclinical development, anticipating that we'll initiate studies in healthy volunteers in the very beginning of next year. We can move rapidly through these single dose studies in healthy volunteers and then advance into patients with multiple ascending dose studies, getting quickly, hopefully, to doses that will have a real meaningful impact in patients. Our strategy after that is to move into an open label extension and use this data as confirmation of the efficacy and safety of our approach and of ED51 and its ability to help exon 51 skippable patients. Next slide, please. Now, this isn't the only program that we're working on at PepGen. We are actually working on several other exon skipping approaches to be able to broaden our ability to treat patients with Duchenne muscular dystrophy. We also have a program in myotonic dystrophy where we think our approach and our ability to go and target different muscle groups efficiently and safely will allow us to provide real benefit to these patients. And in particular, our ability to access the CNS may be of tremendous benefit for those patients as well. Beyond that, we have plans to go and expand across different neuromuscular diseases to try and bring therapeutic benefit to a broader group of patients who are desperately in need of real meaningful therapies that will change lives. Next slide, please. And with that, I'd like to thank you for your time And I'm happy to take any questions you have.
Sure, thank you so much, James. Just a quick question. We heard from the two companies that are using a form of an antibody to help with delivery, and now you're talking about a peptide to enhance delivery. Can you explain to the community more about that and talk about that peptide?
Terrific. Yes, so this is the work of our two academic founders that the company then took several steps beyond that to go and create a really efficient tool to get into muscle cells. So it doesn't use a receptor, unlike the approaches that were described already, but uses a non-specific mechanism to get into muscle cells, but to do so very efficiently. Now, one of the advantages we think of this approach is this is a very small addition to the oligonucleotide. And so we think it'll get really good exposure deep into tissues, deep into the heart, deep into muscles. And we think that allows more oligonucleotide to get into cells, particularly deeper into the tissue. When we go and look at muscles in different animals, we can go and see that we get very good distribution throughout the tissue. And we think that's critically important for this disease.
Great. Thank you, James. I appreciate that. Now I'm going to turn it over to Liz from Sarepta Therapeutics. Liz?
Thank you, Ryan and Eric, and thank you to the audience for joining us today. My name is Liz Smith, a nurse practitioner and interim head of U.S. Medical Affairs at Sarepta. Today, I'll be sharing updates on our PPMO exon skipping program. Next slide, please. Please take a moment to view our forward-looking statements on our webcast. Some of today's updates will be forward-looking, meaning they haven't yet happened, and there's a risk and uncertainty associated with these statements. Next slide, please. Sarepta's experience in Duchenne runs deep, with over a decade of dedicated research. To meet our mission of reaching 100% of individuals living with Duchenne, we have three diverse approaches to Duchenne drug development: RNA-targeted therapies, which is exon skipping, gene therapy, and gene editing. We have several programs that span our RNA platform using our PMO and PPMO technology, including 3 exon skipping products that have received accelerated approval in the United States. We have an active microdystrophin gene therapy clinical program. We have gene editing partnerships in early stages of development. For this presentation, we're going to focus on our PPMO platform. For a more detailed presentation, please see our PPMO pre-recorded presentation in the conference's on-demand library. Please also see our pre-recorded presentations on our PMO pipeline and gene therapy program in the PPMD on-demand conference library. Next slide, please. In the presentation today, I'm going to focus on our PPMO investigational product, SRP-5051, which is currently in clinical stage. We have previously completed an early stage study called a single ascending dose study. Today, I will focus on Momentum, our multiple ascending dose early stage trial for the evaluation of SRP-5051, our first PPMO product in clinical trials. Momentum is a 2-part multiple ascending dose study for ambulatory and non-ambulatory individuals ages 4 through 21. This slide shows a study schematic for Part A of Momentum. The goal of Part A was to evaluate the safety and tolerability of SRP-5051 at multiple ascending dose levels in order to determine the appropriate dose for further advancement. Patients received one of the escalating doses of SRP-5051 shown on this slide, and dosage increases occurred after clearance by the Safety Review Committee. Unlike PMO, which is weekly dosing, SRP-5051 was administered once every 4 weeks, so patients in the trial received dosing every 4 weeks via IV infusion for at least 12 weeks. Today, we'll be sharing data for 4 patients in the 30 mg/kg cohort of Momentum. We will be sharing data from this cohort because this is a dose that we will bring to the FDA to discuss as a target dose to bring into Part B of the trial. Part B is anticipated to be the pivotal trial pending FDA engagement and endorsement. 3 patients were in their late teens and non-ambulatory, and 1 patient was 7 years old at the time of treatment. Next slide, please. To understand if SRP-5051 is operating as intended, there are 2 questions that we ask. Was exon skipping observed, and is dystrophin protein produced after SRP-5051 administration? SRP-5051 dosed monthly at 30 mg/kg achieved 10.79% mean exon skipping at week 12. Compared to PPMO 20 mg per kg dose, there was a greater than 4-fold dose-dependent increase in exon skipping at only a 50% increase in dose. The data also showed that the increase in exon skipping resulted in an increase in dystrophin. The 30 mg per kg dose of SRP-5051 resulted in a 6.55% mean dystrophin protein expression as measured by Western blot. We hypothesized that dystrophin production will increase over time. So we were encouraged that even at this early time point, the data represent a greater than 100% increase in exon expression versus the 20 mg per kg cohort. Next slide, please. Now, I'll talk a little bit about safety. This slide shows the safety experience across all studied doses of Momentum. In the 30 mg per kg cohort, 2 patients experienced what we classified as a serious treatment-related adverse event, which we'll explain more in detail. Patient 1 experienced 2 serious treatment-related adverse events, and patient 3 experienced 1, resulting in 3 total in the 30 mg per kg cohort. 2 cases were of reduced magnesium or hypomagnesemia, and 1 case was of reduced potassium or hypokalemia. None of the serious adverse events were life-threatening, and both patients remained asymptomatic. Next slide, please. So to summarize, in the 30 mg per kg cohort, we detected mean exon skipping of 10.79% and mean dystrophin expression at 6.55%. The patient receiving the most SRP-5051 doses, 5 versus 3, at the time of biopsy had the highest expression. Their exon skipping was 11%, and dystrophin production measured by Western blot was 12.1%. We hypothesize that exon skipping and dystrophin will increase with longer-term dosing. Future clinical studies will test this hypothesis. In terms of safety, hypomagnesemia is a newly identified risk in the SRP-5051 program. Most cases were mild and asymptomatic, with some serious cases occurring prior to the implementation of serum monitoring and magnesium repletion. Our analysis so far indicates that hypomagnesemia or low serum magnesium is manageable and monitorable. Markers of kidney function have generally been normal and have not shown any consistent relationship to the hypomagnesemia. In addition to our analysis, we've consulted with external expertise and believe that serum monitoring of magnesium and oral supplementation with magnesium is a feasible approach to enable early detection and management going forward. Our plan is to meet with the agency to discuss this data and plan for next steps in the trial. Next slide, please. For the sake of time, I won't go into detail, but you can find more information on our pre-recorded presentation. We are committed to advancing our PMO and PPMO therapies and supporting the patients receiving these therapies. We are continuing to use data collection to inform next steps for subsequent exon skipping trials. And we want to thank you, PPMD, for having me here to speak today. And I look forward to being able to respond to some questions. Of course, if you have any questions, please don't hesitate to reach out to our patient affairs team at advocacy@sarepta.com. Next slide, please. Thank you, and back to you, Ryan and Eric.
Thank you so much. Just one quick question before we open the panel. When can we expect to see some of the longer-term expression data to get a better sense of the average level of expression? I know timelines are tough, but we always got to ask. It's our responsibility, so.
Yeah, so more to come on that hopefully sooner than later. We're working with the agency now to regarding timelines for restarting after the hypomagnesemia occurred.
Thank you.
Great, thanks. So yeah, we're going to move to the open Q&A. So if you have questions, again, please submit them in the box below. And so actually one of the first ones we have in is for Sarepta. So obviously you're working with this PPMO strategy. It's kind of a two-parter. So if it's successful, and you're able to take it forward and get approved, is this something with your already approved products for 51, 45 that would all be transitioned over to this PPMO strategy?
So for right now, our goal is at Sarepta to advance. Right now, at this time point, we don't have a specific timeline for additional exon skipping therapies. We're learning from our 5051 Momentum and our Mission High Dose study. And as we get more information, that will help inform the direction moving forward.
Okay.
It's an important question that we are actively trying to address.
Yeah, because the follow-up question we had was from a family specifically about, one of the ones that's been in the pipeline for a little longer, like skipping exon 52, and they were wondering, is this something where we're going to have to wait and see an approval for the PPMO before that would get targeted, or if that's going to be, like you said, maybe based on what you learned from the mission study and the high dose about how that goes, essentially.
So, as I said, we're waiting to inform based on the results of mission and momentum as we expand into the other exons.
Great, thank you.
But our plan is to, continue to develop and expand into other exons. Thanks.
And then, so we saw some data from PepGen looking at some of that distribution and exon skipping in non-human primate models. For Avidity and Dyne, are you thinking about looking at other models besides just the mouse before you enter into your clinical trials? And this could be for all three of you. Is there any interest in looking at some human tissue before you kind of make that next step to first in human? So I'm going to start with you, Ash, because you're at the top of my screen.
Yeah, sure. So in order to, so the answer is yes. We will be looking at non-human primates. It's important data to collect in this particular model to look at the type of data that you saw in terms of tissue distribution and skipping. For those who may not be aware, there is not a dystrophic model, a dystrophic non-human primate model. So you can't look at dystrophin production per se. These are healthy, non-human primates. But it does give you very important tox information and other information that you would want to collect. And so we are planning on those studies, and those studies feed into what we would call an IND, investigational new drug application, that we submit to the FDA so that we can start our clinical program. So the answer is yes. In terms of patient cells, just very briefly, we have done that study, in vitro studies in patient-derived cells. I didn't show it here, but we do have some of that data on our corporate website.
Great, thank you. And Lisa, for Avidity?
Yeah, great. It's basically the same answer. Non-human primates are a really important bridge species, especially with oligonucleotide therapies. They've been shown to be very good for investigating the PK and PD properties of these oligo drugs. So absolutely, we are doing non-human primates, and it will be part of our package for our IND as well. And given, just as Ash said very nicely, given that there's no dystrophin models for the exon skipping that we're looking at that are easy to get to, we have really relied on human myotubes from patients who have donated their myoblasts, and we've done a lot of experiments showing that our different exon skipping drugs work in these MDM-derived myotubes. So yeah, that's also a really important piece of our package.
Great, thank you. James, any additional data you're looking to collect before starting that first? And I believe with yours is going to be an adult population, correct, for the same study?
Yeah, I mean, Ash and Lisa are correct. You can't look at dystrophin production in these models, but in addition to safety, you really get some extraordinary data in terms of biodistribution and also efficacy, because you can look at exon 51 skipping. And obviously more exon 51 skipping results in more dystrophin protein. That's how all of our drugs work. So we think actually it's actually a critical indicator to say, is this really going to move the needle in a really fundamental way for patients? So for us, we think this is the next step that we've now completed. We are finalizing our toxicology package that will be part of regulatory filing. And with that, we look forward to actually getting data next in humans and moving as quickly as possible and as safely as possible into children with DMD.
Great, thank you.
So I'm going to ask the question that we hear so often from the community. Why do we always start with exon 51? It's a crowded field. There's a lot of activity. It's always targeted. The community needs to know, is it a business decision? At the end of the day, we have patients waiting. And I'm also worried about even recruitment. We're going to have to go outside the US possibly. So I think the community deserves to know an answer to this. So I'm going to start with Elizabeth Ackerman. Put you on the spot first.
Well, actually, good news is we aren't starting with exon 51. We're starting on with exon 44, which of all the exon amenable patients, that's, I think, has been one of the most underserved in the community. So we are super excited that we're going to be able to have our first drug in DMD be for that section of the community.
Great, but 51 will be followed by that before the rarer exons, correct?
We're starting with 44, and that's followed by 45 and 51.
Correct. Okay. So it's still in the beginning of it, and we are grateful that 44 is being targeted. And I want to stress that that is an underserved space, but the rare ones are still followed down the road. Okay, Ash, I'm going to turn to you.
Sure. So it's a great question. There's still huge unmet need in all of these, with all of these mutations. 51 is the biggest population, and it's still, there's still a lot of need for improvement, frankly. And so, we are targeting 51 first, with 53, 45, 44 behind that program. And you're right. It is a crowded landscape for clinical trials as well, and we are going to be conducting a global development program for our 51 development.
Great, James.
Yeah, first let me just say kudos to Lisa and Avidity for going into 44 first. I think that's terrific. And just to follow on what Ash said, we think that the trailblazing that Sarepta has done is extraordinary, and we're all looking to do better. And we really think at PepGen that our approach really can do something extraordinary for patients. If we could produce double-digit levels of dystrophin protein after 3 months of therapy, that would be a game changer. So why start off in 51? For us, actually, it's that there's so much validation, if you will, in terms of the clinical development path, the outcome measures. And for us and for investors, we find that it's easy to go and say we can benchmark ourselves against those who have gone before us and say, how much better are we doing? And that allows us to then really drive forward into other approaches. One of the great things, though, about this peptide-based approach is very rapidly, we can go and do a plug-and-play approach, take the same peptide and put it on a different oligo and move from one indication to the next indication to the next indication. And that's what we're building the team to go and do. We think this is what the community deserves.
Yeah, just as maybe a follow-on to that, so since there is some overlap in terms of the exons that we're targeting, and there's going to be, there's only so many patients with those mutations, and we do see a lot of trials kind of target that same early ambulatory 4 to 7 range. You're probably early in your discussions around clinical trial design, but are you thinking about other populations that maybe don't have or aren't given the opportunity to participate in these trials as early? As a way to kind of power your studies. So James, I'm maybe going to throw that back to you real quick.
Yeah, I think that's a great point. And I think we do need to go and broaden how we're looking at patient populations and accessible patient populations and patient populations where we can learn a tremendous amount clinically. And we're fortunate that Jane Larkindale has joined us at PepGen and is a great thought leader in terms of thinking about how do we do those sort of trials and get to those sort of patients and provide a broader benefit. Great question, though.
Great, thank you. No, and we miss Jane already from her work in de-risk, so good.
Oh, she hasn't left you.
No. We know where she lives, so.
And we actually have some other questions coming in, so I think I'm just going to kind of bounce around real quick. So, Liz, with Sarepta seeing kind of the low magnesium levels in response with treatment, is that something, you talked about kind of adding that to the protocol, is that something patients should be maybe expecting? Is that something we're just going to see throughout, the long-term treatment with this drug that becomes kind of part of that process of the infusion that you're going to be have some additional, treatment layered on there?
So for part of the clinical trials, we will be adding additional monitoring for magnesium to monitor for hypomagnesemia. We believe that this is monitorable and manageable. We've sought out expert review from nephrologists, but we will be continuing to monitor in the ongoing clinical trials, and we'll know more as we progress in Part B for momentum.
Thank you. And then it's early, obviously, so hard to tell some of this information with your products in Duchenne. But questions about these other ASOs using conjugates, potential safety or side effects, or the things we can learn from other indications or other places where there's monoclonal antibodies being used or the Fabs being used that we can maybe take as you kind of are planning your trials as well. And Lisa, maybe I'll start with you.
Okay, yeah, great question. And I think, monoclonal antibodies have been used now for a long time. There's a lot of drugs out there. We know how to make them. We know how to make them safer. So it's in the same thing with oligonucleotides. So we're taking two very well-known, well-used modalities that are already used in many drugs that are out on the market and hooking them together. So of course, when you hook them together, that makes it new and you never know what you're going to find. But at least the two pieces we have a lot of confidence in. And we also, by using the same antibody for all of our neuromuscular programs, what we learn from one program will really be helpful for the other program. So we will be going into the clinic for DM1 using this monoclonal antibody that we're using for DMD. So that was going to be really important to form us on the safety. But so far, all of our non-clinical data looks very good.
Great, thank you. Ash, if you'd like to add anything.
Yeah, just a little bit. So, as Elizabeth said, there is a good deal of information out there on antibody drug conjugates. So I'm sure all companies are looking at what information is in the literature and medicines, different antibody drug conjugates that are on the market now. At Dyne, we're taking a slightly different approach, or a very different approach in terms of taking a portion of that antibody, and we think that'll confer certain advantages. So we're excited to see how our conjugate performs as we move further along. And as Elizabeth alluded to, we have a very modular approach, and so we can interchange PMO that skips 51 or PMO that skips other mutations, which allows us to move our programs through non-clinical development very quickly. And then we are also focused on other diseases like myotonic dystrophy type 1 and FSHD, where our Fab and linker remain constant. And it's that therapeutic agent that we can interchange. And so we're able to move our programs through very quickly because of the modularity of the FORCE platform.
Great, thank you. And I just want to say thank you to all the panelists. We appreciate Sarepta sharing some of your data, and with the other companies, talking a little bit about what you've been working on to date, and kind of looking forward to your clinical trials as you all advance your program. So look forward to speaking with you all in the future.